Hello everyone, I'll keep this introduction short and sweet. This is the first video I'm uploading on nitrogen biochemistry. It's an introduction to DNA. We're looking at DNA purely from a biochemical aspect. We're not looking at cellular location in cells. We're not looking at chromosomal arrangements or histone interactions. They'll be covered in later videos. For now, we're focusing on advanced biochemistry for life science students, including biochem, molecular biology, pharmacology, and biological science students, as well as medicine students, to help you understand the fundamentals of biochemistry for the purpose of exams. So as I mentioned before, we're purely focusing on the biochemical aspects of DNA. So by the end of this will be uploaded in a two-part video. By the end of part one, you should be able to distinguish purines and pyrimidines on a structural and atomic level, to distinguish the difference between nucleosides and nucleotides, and to understand nucleosides' fate in metabolism, as well as to understand the atomic structure of the sugar phosphate backbone and its role in the polymerization or forming a DNA polymer. The next video that will be released will go over hydrogen bonding, how it mechanistically plays a role in base pairing, and how hydrogen bonding contributes to the stability and shape of the DNA double helix, as well as predicting the directionality, 5' to 3' and chemical stability of DNA. So what is DNA? Purely from a chemical perspective, DNA is considered a polymer. Now, in chemistry, a polymer refers to a large chain molecule that's made up of many small repeating chemical units. And these chemical units are referred to as monomers. So many monomers will make up a polymer. Now, a DNA polymer is referred to as a polynucleotide, and a polynucleotide is made up of many monomers, and they're referred to as mononucleotides in the context of DNA. Now, if you were to take one of those mononucleotides, you would see a nitrogenous base, a deoxyribose sugar, and a phosphate group. Now, the nitrogenous base is indicated in purple, and that can either be a purine or a pyrimidine. The deoxyribose sugar is a pentose sugar, and that links the nitrogenous base through a glycosidic bond. The phosphate group is linked to the deoxyribose sugar through a phosphoester bond. Many of those repeating chemical units will link together to form a polynucleotide. The only difference is the units may differ, the mononucleotides may differ in the nitrogenous base. So that will either be adenine, guanine, cytosine, or thymine. So just to reinforce and to build a better picture, a DNA polymer is referred to as a polynucleotide. It's made up of many mononucleotides, okay, and they're repeating chemical units. And you can see here I've got a better example on the left of what will start to look like a DNA polymer. So we have our 4 different nitrogenous bases. You can see that the difference in the repeating chemical units is the nitrogenous bases that are linked to the deoxyribose sugar. It's still a glycosidic bond for every single base, and the deoxyribose sugar is linked to the phosphate through a phosphoester bond. But you can see that that phosphate group is actually linking the deoxyribose sugar on the other nucleotide monomer. So therefore, in a polynucleotide, the backbone, if you like, is referred to as a phosphodiester bond, okay? But that phosphate sugar backbone is what is pretty much consistent, or what is consistent across the structure of a DNA polynucleotide. The nitrogenous bases the deoxyribose sugar is linked to will differ, but it will either be adenine, guanine, cytosine, or thymine, which we're going to look at in the next slide. But before we move on to that, I just want to kind of reinforce this kind of concept of DNA as a polymer and why it's a very unique polymer. So you can see glycogen, as you're aware, glycogen is stored in the liver. Okay, glycogen is also a polymer. It's a polysaccharide, right? And it's made up of many monomers referred to as monosaccharides, and they're individual glucose molecules, if you like, and they're all bonded together via a glycosidic bond, okay, and an oxygen bridge. But you can see the predictability, there's some level of predictability and structure here, right? Any kind of unit you take out will be glucose. It will either be a monosaccharide or a disaccharide, which would be 2 glucoses linked together. But either way, at its core, it's just glucose. But DNA is a slightly different polymer. You can appreciate its complexity. So yes, the phosphate sugar backbone is consistent, but at any given point, you don't know whether you're going to get a purine base or a pyrimidine base. You don't know if the nitrogenous base could be adenine, it could be guanine, or the pyrimidine base could be thymine or cytosine. So that's just a comparison between different polymers we see in our own biology, glycogen versus DNA. So again, just to reinforce, DNA is a polymer. It's referred to as a polynucleotide. It's composed of many monomers or mononucleotides that are joined together to essentially form a large, long strand molecule. Okay, so that within a DNA polymer, you have a mononucleotide, and that will consist of the nitrogenous base. It's bonded to a deoxyribose sugar via a nitrogen bridge. It was called a glycosidic bond, and then that deoxyribose sugar is bonded to a phosphate group via a phosphoester bond. Now, the nitrogenous bases, as I mentioned before, there's 4 nitrogenous bases that make up DNA altogether. Okay, so what are nitrogenous bases? So nitrogenous bases, they're heterocyclic aromatic molecules containing nitrogen, and they participate in hydrogen bonding and electron delocalization, which is very, very important. When we later on in the video, when we look at the DNA double helix structure and the stability of DNA, it's the chemistry of the nitrogenous bases is crucial in keeping DNA as its structure. Okay, now what do we mean by heterocyclic aromatic molecules? Now, if you've done organic chemistry, you know that aromatic molecules are ring-like structures, okay? And heterocyclic just means they're made up of more than one atom in that core ring. So for nitrogenous bases, their core ring consists of carbon and nitrogen. Okay, and again, we're going to go over hydrogen bonding later on in the presentation. For now, just know that they participate in hydrogen bonding. Okay. Now, these nitrogenous bases, they can be divided into 2 groups, 2 families, if you like, purines and pyrimidines. Okay, so as I said before, purines can include guanine and adenine. Pyrimidines include cytosine, thymine, and I've mentioned here uracil. Now, we're not really going to be talking about uracil too much in this video, but uracil is actually present in RNA. It's not present in DNA. Okay, and thymine, conversely, is present in DNA, but it's not present in RNA. And we're going to get into all that later. You'll understand why. But just know for now that nitrogenous bases, they're heterocyclic aromatic molecules, which means they've got more than one atom that make up that core ring-like structure. So the first family of nitrogenous bases we'll cover is the purines. Now, purines consist of adenine and guanine only. So you can see they're both made up of 2 aromatic rings, one pyrimidine ring and one imidazole ring. So the pyrimidine ring is 6-membered and the imidazole ring is 5-membered. All that means, if you did organic chemistry, is, for example, imidazole has 5 atoms that make up that core ring-like structure. But it's important that both adenine and guanine, they're still heterocyclic, and both the pyrimidine and imidazole ring is heterocyclic in nature. So it's not just carbon-based, nitrogen is also incorporated. More than one atom is making up that core ring-like structure. Now, the main difference in molecular structure is their functional groups, okay, and I'll go over functional groups a lot more in part 2 when we look at hydrogen bonding. Okay, these functional groups are integral in governing that kind of complementary pairing you see in the DNA double helix structure. Okay, the nature of functional groups themselves are incredibly important across biochemistry. So I am going to go into them in more detail, but for now we're just going to look at them as we see on paper. Okay, so the functional groups are different in adenine and guanine slightly. So on that 6th carbon on the pyrimidine ring, that very top carbon on the 6-membered ring, the functional groups are different. So in adenine, it's nitrogen bonded to 2 hydrogens, which is an amine group. Okay, so that on that 6th carbon, it's got an amine group. Whereas on guanine, that 6th carbon on the pyrimidine ring, it has a carbonyl group, C double bonded to an oxygen. Guanine also, however, has an amine group on its 2nd carbon. You can see it's still got that nitrogen double bonded to, it's that nitrogen bonded to 2 hydrogens, not double bonded, bonded to 2 hydrogens on its 2nd carbon, whereas adenine just has a hydrogen. Okay, and you can also see they're slightly different in their ring arrangement. The bonding is slightly different, and that's because of the functional group. You can see in guanine, so on the 6th carbon of guanine, you have that carbonyl group. It's essentially taken that bond So it's just a single bond between that carbon and nitrogen, whereas in adenine it's lacking that hydrogen and it's also lacking that carbonyl group. So the double bond is between that carbon and nitrogen. Now, in an exam-like situation, you may be asked to contrast and compare the structures, and they're very easy marks. So in the sense that we can compare, they're both heterocyclic rings. They both have a 6-membered pyrimidine ring and a 5-membered imidazole ring. Both rings are heterocyclic. They both have an amine functional group. However, the amine functional group is located on different carbons respectively. In adenine, it's on the 6th carbon, and in guanine, it's located on the 2nd carbon. And guanine has an additional functional group. It's got a carbonyl group. Adenine does not have a carbonyl group, and its carbonyl group is on the 6th carbon on the pyrimidine ring, whereas adenine has an amine group on the 6th carbon on the pyrimidine ring. Now, what we're looking at here is essentially a free nitrogenous base, so it lacks that phosphate sugar linkage. Okay, so when we looked at the nucleotide monomer earlier on, you saw the nitrogenous base bonded to a deoxyribose sugar via a glycosidic bond, and that deoxyribose sugar was bonded to a phosphate group via a phosphoester bond. Now, it's important to know that this glycosidic bond with the deoxyribose sugar in purines is always formed on the 9th nitrogen, and that 9th nitrogen is located on the imidazole, the 5-membered ring you see. So that nitrogen right down there that's got that hydrogen sticking out of it, that's where the glycosidic bond will form between the deoxyribose sugar. So again, just to recap, purines have 2 rings, they're heterocyclic in nature, and they differ in functional groups. It's got an imidazole ring, it's got a pyrimidine ring, and it's got guanine has an additional carbonyl functional group, and the amine group is located on different carbons respectively. So we've just spoken about purines, and now we're going to speak about the other family of nitrogenous bases, which are pyrimidines. Okay, now pyrimidines are slightly different in their structure. As you can see, they don't have that imidazole ring, that 5-membered atom ring. They only have a pyrimidine ring, which is a 6-membered ring, but the ring is still heterocyclic, so it's important you can see that it's incorporated nitrogen as well. It's not just carbon, it's composed of carbon and nitrogen, covalently bonded. Okay, and that's the heterocyclic ring. Now, you can see that thymine, uracil, and cytosine are the 3 pyrimidines that you need to know. But uracil is not present in DNA. It's really important to remember that. But it's present in RNA. And conversely, thymine is not present in RNA, but it's present in DNA. And it's interesting, actually, because from looking at their molecular structure, if you notice, they're almost identical. The only difference is that thymine contains a methyl group on its 5th carbon, whereas uracil doesn't. Now, thymine can be synthesized from uracil by thymidylate synthase, and using folate as a, I guess, cofactor to kind of stabilize that reaction, and it becomes methylated. So the methylation of uracil, you will get thymine. Okay, so when looking at their molecular structure, you can kind of take 2 birds with 1 stone, and you can say thymine and uracil, they have an identical structure, only thymine has that methyl group on its 5th carbon. Okay, so they're almost identical. Now, in terms of functional groups, both thymine and uracil also have a carbonyl group on their 4th carbon, but all 3 pyrimidines have carbonyl groups on their 2nd carbon. So again, I mentioned when we were talking about purines, the carbonyl group is expressed in guanine, but it's not present in adenine. That carbonyl group is the C double bond O, okay, and that's a functional group. Now, cytosine has an amine group on its 4th carbon, and thymine has a methyl group on its 5th carbon. Okay, but in terms of, again, same questions with purines, with pyrimidines, they may ask you to compare and contrast. So if we were to compare and contrast, we can say all of them contain carbonyl groups in their 2nd carbon. Cytosine lacks that carbonyl group on its 4th carbon. Instead, it has an amine group on its 4th carbon. Thymine and uracil are pretty much similar in molecular structure, only thymine has a methyl group on its 5th carbon. Now, I mentioned before that sugar linkage, that phosphate sugar linkage is really important in polymerization, but also in forming a nucleotide itself. Okay, so that sugar actually links to the 1st nitrogen. Okay, so the nitrogen at the very top, okay, that glycosidic bond with the deoxyribose sugar is formed with the nitrogen at the very top. In the case of uracil, it's the same principle, only in RNA you don't have a deoxyribose sugar, you have a ribose sugar. Okay, so that first nitrogen forms a glycosidic bond in uracil with a ribose sugar, but for thymine and cytosine, it's that first nitrogen, the glycosidic bonds happens with a deoxyribose sugar. Okay, so just to recap, pyrimidines, much smaller, they lack that imidazole ring, they have a six-membered pyrimidine ring, it's heterocyclic still, composed of nitrogen and carbon. Both thymine and uracil have carbonyl groups on their second and fourth carbon respectively. However, thymine has an additional methyl group on its fifth carbon, and cytosine has a carbonyl group on its second, and instead of having a carbonyl group on its fourth carbon like uracil and thymine, it has an amine NH2 functional group on its fourth carbon. So that's just basically an overview of purines and pyrimidines and their atomic structure. So just to recap and to look at the bigger picture, if you like, we've just looked at the three nitrogenous bases. We've looked at purines and pyrimidines as free bases themselves, right? Just the nitrogenous bases without that phosphate sugar linkage. Is that phosphate sugar linkage that is a mononucleotide? Okay, so you can see this is an example of an adenine monomer or an adenine mononucleotide. So you can see you've got the nitrogenous base, you've got its amine functional group on its sixth carbon, and you can see the imidazole ring, that ninth nitrogen, is forming a nitrogen bridge with a deoxyribose sugar, and this is referred to as a glycosidic bond. And just to get familiar with terms, a glycosidic bond is either a bond between two sugars or it's a bond between a sugar and another molecule. And then furthermore, you can see we've got the phosphate group in the back, and that's linked to the deoxyribose sugar via a phosphoester bond. And that is our nucleotide monomer, essentially. So you can kind of see where it fits into the whole picture. And many of those nucleotide monomers, whether the nitrogenous base is guanine, cytosine, or thymine, or adenine, will polymerize together and form one long strand of DNA, a polynucleotide. So just to reinforce and recap, we can now distinguish purines and pyrimidines on a structural and atomic level. Okay, for example, we know that purines consist of two aromatic ring-like structures, one six-membered pyrimidine ring and one five-membered imidazole ring. Okay, whereas pyrimidines only have that six-membered pyrimidine ring. Okay, both purines and pyrimidines are heterocyclic in nature, which means that more than one atom makes up those core ring-like structures. Okay, so in both purines and pyrimidines, it's carbon and nitrogen respectively. Okay, it's also important to remember that in purines, both the imidazole ring and the pyrimidine ring is considered heterocyclic. So the imidazole ring is composed of nitrogen and carbon, and the pyrimidine ring is also composed of nitrogen and carbon. Okay, we know the main difference between the two purines and between the three pyrimidines on a molecular level is their functional grouping system. Okay, and this is what dictates or governs the hydrogen bonding in the complementary pairs later you see in DNA. Okay, so for example, in guanine, the sixth carbon on the pyrimidine ring, it possesses a carbonyl group, C double bonded to an oxygen, so carbon double bonded to an oxygen. In adenine, that same sixth carbon, instead of having a carbonyl group like guanine, has an amine group, so nitrogen bonded to two hydrogens. It's also important to remember that guanine has that additional functional group. On its second carbon in the pyrimidine ring, it contains an amine group, so nitrogen bonded to two hydrogens, whereas adenine on that second carbon in the pyrimidine ring only has a hydrogen. Okay, so guanine has an additional functional group. Okay, we know that pyrimidines only consist of a six-membered pyrimidine ring. They can either be thymine, uracil, or cytosine. We know that uracil is not present in DNA, but thymine is present in DNA. We know that uracil is present in RNA, but thymine is not present in RNA. We know in terms of molecular structure, thymine and uracil are very, very similar. They both possess carbonyl groups on their second and fourth carbon within the pyrimidine ring. Okay, the only difference is thymine has a methyl group on its fifth carbon, and it's the only nitrogenous base to possess a methyl functional group. Okay, cytosine also possesses a carbonyl group on its second carbon. Okay, but instead of having a carbonyl group on its fourth carbon like thymine and uracil, it possesses an amine group, so an NH2, nitrogen bonded to two hydrogens. Okay, so now there's differences in phosphate sugar linkage. Okay, so in purines, the phosphate sugar linkage occurs in the imidazole ring. It occurs in the ninth nitrogen, which is located in the imidazole ring. Okay, so that glycosidic bond with the deoxyribose sugar will form with the ninth nitrogen located in the imidazole ring. In pyrimidines, however, that glycosidic bond forms with the first nitrogen in the pyrimidine ring. Okay, so it forms a glycosidic bond with deoxyribose sugar with the first nitrogen in the pyrimidine ring. It's important to remember though that uracil forms a glycosidic bond with a ribose sugar and not a deoxyribose sugar because it's only present in RNA. It's not present in DNA. Okay. Same with thymine. Thymine does not form a glycosidic bond with a ribose sugar because it's not present in RNA. So it will only ever form a glycosidic bond with a deoxyribose sugar. So thymine is, and that's on the first nitrogen. So that's essentially a comparison and a contrast and comparison between those nucleotide or nitrogenous bases. So we've previously just spoken about nucleotides, and now we're going to be speaking about nucleosides, okay? And again, it all kind of falls into place, so it's not very complicated. So nucleosides are essentially that bridge between a free base and a functional nucleotide, okay? So they're that bridge between a free nitrogenous base and a functional nucleotide. So we know that a functional nucleotide consists of the nitrogenous base, right? It's bonded to the pentose sugar via a glycosidic bond, and that pentose sugar links that phosphate via a phosphoester bond, okay? And that nucleotide is able to polymerize, that mononucleotide can polymerize and form DNA, which is a polynucleotide, okay? So for this reason, nucleosides, because they lack that phosphate group, they're referred to as biologically inactive. Okay, so they're metabolic intermediates. So nucleosides can become nucleotides. Okay, this is almost like the kind of pre-step, if you like, before DNA polymerization happens. However, their metabolic fate is quite flexible, right? It's determined depending on the cellular needs. So nucleosides can become nucleotides, but they won't necessarily become nucleotides. They won't necessarily become nucleotides for the purpose of DNA polymerization, okay? Some of them are salvaged, not just for nucleic acid synthesis, but for example, as we'll go on and progress with videos, we'll cover more examples of this, but like GTP, for example, guanosine triphosphate, it's really important in signaling pathways, like MAPK signaling pathways, okay? So these nucleosides have a very flexible fate, okay? And another difference between nucleosides and nucleotides is their name. So a guanine nucleotide is called guanine, right? But when it lacks that phosphate group, when it's a nucleoside, it's referred to as guanosine. Okay, and the same with adenine. Adenine then becomes adenosine, right? Cytosine becomes cytidine. Uracil becomes uridine, and thymine becomes thymidine. So again, just to recap, nucleotides are functional. They're able to polymerize and form a polynucleotide, okay? They're able to form DNA. Nucleosides are not functional. They lack that phosphate group, okay? They are just a nitrogenous base bonded to the ribose sugar via a glycosidic bond, okay? So it's just the base and the pentose sugar. So as I said before, nucleosides themselves are biologically inactive. However, they can become DNA-ready nucleotides. So they can become biologically active and ready to polymerize and form a polynucleotide or a strand of DNA. But this process is a stepwise phosphorylation process, and it's specific kinase enzymes are required for this process to happen, right? They catalyze this chemical reaction. Okay, and altogether 3 phosphate groups are added to the nucleoside. But just to recap, just to avoid confusion, right, a mononucleotide only has 1 phosphate group, okay? But in order for the polymerization to happen, a beta and gamma phosphate must be cleaved from the nucleoside triphosphate in order to allow for that, in order to provide the energy for that polymerization to occur, okay, which we're going to talk about in the later slides. But for now, just understand that in order for a nucleoside to become a DNA-ready nucleotide, we have to phosphorylate it 3 times to get a deoxyribonucleoside triphosphate, and that's usually referred to as a DNTP. So I'm going to go over each process in a lot more detail in the coming slides, but for now I've just done a table to kind of summarize the first step. Okay, the first step is phosphorylating our deoxyribonucleoside into a DNTP MP. That's a deoxyribonucleoside monophosphate. Okay, so just to break the language down, we start off with our deoxyribonucleoside, for example, deoxyadenosine, right? That's just the adenine nitrogenous base bonded to the deoxyribose sugar through a glycosidic bond. There is no phosphate, okay? But the enzyme deoxyadenosine kinase, with the help of ATP and magnesium cofactor, will help phosphorylate that, and it will become deoxyadenosine monophosphate. Okay, so we've got deoxyadenosine monophosphate. Mono, there's one phosphate group now. One phosphoester bond has formed between that pentose sugar and that phosphate group. Okay, and it's the same principle for deoxyguanosine, deoxycytidine, and thymidine. Okay, their respective kinase enzymes, along with ATP as a phosphate donor and magnesium as a cofactor, will help catalyze this and bring this reaction together, right, to give us deoxyguanosine monophosphate, deoxycytidine monophosphate, and thymidine monophosphate. Okay, so that's step one complete. So step one is going from a nucleoside, a deoxyribonucleoside, to a deoxyribonucleoside monophosphate. As I mentioned, it's a three-step phosphorylation process. So we're still at step one, but I just thought I'd break it down a little bit more so we can understand what's happening on a chemical level. Okay, so here we start off with our deoxyadenosine. So as I said, it's just a nitrogenous base. It's linked to the deoxyribose sugar via a glycosidic bond. Okay, that's our nucleoside. Okay, it's biologically inactive, and it's that bridge between a free base and a functioning nucleotide. Okay, now ATP is adenosine triphosphate, as everybody should be familiar with ATP. Okay, now ATP in this case is our phosphate donor, so it's gonna donate its gamma phosphate, which is the third phosphate on the ATP, and I'll represent that in a diagram later. It's gonna donate its gamma phosphate to the deoxyadenosine. Okay, but it needs the help of magnesium to do that. Magnesium acts as a cofactor, and it stabilizes. It acts as a charge shield, and I'll explain that as well in another slide later on. Okay, so we've got our deoxyadenosine plus ATP plus magnesium. Okay, and we need our deoxyadenosine kinase. Now, I'm not the best at PowerPoints, so I typically in an equation the enzyme is shown above the arrow, but I couldn't, for some reason it didn't want to do that, so I've just highlighted in yellow deoxyadenosine kinase. But also an important point as well that reminds me is DCK. Okay, deoxycytidine kinase in certain cells will act as a universal phosphorylator, so it's able to phosphorylate not just deoxyadenosine but deoxyguanosine. It's quite confusing, but I'll get into that later. But anyway, this gives us deoxyadenosine monophosphate. So you can now see that hydroxyl group has now linked that phosphate group. We've got a phosphoester bond here. We've got our deoxyadenosine monophosphate. So this pretty much looks like a nucleotide monomer, right? We've got our nitrogenous base, the adenine nitrogenous base. It's linked through a glycosidic bond with the deoxyribose sugar, and that deoxyribose sugar is linked with a phosphate via a phosphoester bond. Okay, and I'll talk about the role of magnesium as well later, but magnesium is not used up or consumed in this process, right? It's a cofactor. Now my little icon is actually covering up the rest of the equation, but as a byproduct we get adenosine diphosphate, right? And that makes sense because ATP is donated a phosphate. Okay, so ATP is the one that's out of these two. ATP is the one that's coming out unchanged, along with the deoxyadenosine. So we get our deoxyadenosine monophosphate and we get adenosine diphosphate as a byproduct. Okay, but magnesium is not indicated as a byproduct, not because it's not used up, it's not consumed. It just acts as a stabilizer. Okay, it's stabilizing the negative charge, the repulsion between the beta and gamma phosphates on the ATP and that negative hydroxyl group that's on the deoxyribose sugar, right? It's acting as a charge shield, allowing for the enzyme to kind of bind its substrate. So it's the same principle for guanine, and once again my icon is covering up a key part of the equation, but I've got it here, so I've got it right up here under the title deoxyguanosine. So we've got our deoxyguanosine nucleoside, so that's just a guanine nitrogenous base, and it's bonded to the deoxyribose sugar via a glycosidic bond, right? Okay, and we we take a phosphate from ATP. ATP is generous enough to lend us a phosphate or donate a phosphate, right? With the help of magnesium to stabilize that kind of natural repulsion you'll see between the beta and gamma phosphates on the ATP and that hydroxyl group that's on this pentose sugar here, right? And as a product, we get deoxyguanosine monophosphate and we get adenosine diphosphate. So ATP's lost a phosphate to become adenosine diphosphate, and deoxyguanosine has gained a phosphate to become deoxyguanosine monophosphate. So it's now got one phosphate group. And as I said before, the enzyme that catalyzes this is deoxyguanosine kinase, but it can also be DCK as well. But I think for exam purposes, and to distinguish the step by step, we'll just say deoxyguanosine kinase. Okay. And again, it's the same principle for the cytidine nucleoside. So we start off with our deoxycytidine again. This time we've got a cytidine or cytosine as its pyrimidine ring here. The nitrogenous base is just a pyrimidine ring. There's no imidazole ring. Okay, and it's linked to the deoxyribose sugar through a glycosidic bond. That nitrogen bridge right there. Okay, and this pentose sugar possesses that that negatively charged hydroxyl group. Okay, and that's where we want to form the phosphoester bond. So ATP once again is acting as a donor, it's donating a phosphate, and deoxycytidine kinase is catalyzing this reaction. Magnesium is acting as a charge, as a charge shield again, because there's still that repulsion between the beta and gamma phosphates on the ATP and this hydroxyl group, which is also negatively charged, and I'll get into that more. And deoxycytidine kinase is catalyzing that transfer of phosphates. Okay, and once again, as a product, we have deoxycytidine monophosphates. We've got our 1-phosphate group, and we've got adenosine diphosphate because ATP has lost a phosphate. So again, same principle for thymidine as well. We've got our thymidine nucleoside. It's got there's thymine here as a nitrogenous base. You can see it's got its methyl group on the 5th carbon, right? And then we've got that glycosidic bond, the nitrogen bridge, and we've got this negatively charged hydroxyl group with ATP as a phosphate donor once again, and magnesium as a cofactor or a charge shielder, if you want to call it that, and thymidine kinase catalyzes this transfer of phosphate group. So now this phosphate group appears here, and you get thymidine monophosphate. ATP's lost a phosphate, so instead of being a triphosphate, it becomes a diphosphate. Now, I just want to actually specify something that's quite important here. So you notice that the other nucleosides were referred to as deoxy, whereas thymidine isn't. Now that doesn't mean the pentose sugar it's bonded to, the nitrogen that bonds to the thymine nitrogenous base, right, is a ribose sugar. It's still a deoxyribose sugar, but because thymine isn't present in RNA, it's just assumed that it doesn't exist in that form, right? It is just deoxythymidine. It's just deoxythymidine, whereas guanine, adenine, and cytosine appear in RNA, so they can either form a glycosidic bond with the ribose sugar in RNA or the deoxyribose sugar in DNA. So they just assume, so yes, it's deoxythymidine, but the name's there because it doesn't need to be distinguished. There is no distinction. You just assume when you see thymidine, it will be deoxythymidine. Okay, and again, we've got this monophosphate. We've got thymidine monophosphate. This phosphate group's been added, and ADP is a byproduct because you can finally see it now. My head's moved, but ADP is a byproduct. It's adenosine diphosphate. The ATP's lost a phosphate group. Okay, and magnesium, once again, we're not including magnesium because it's not being consumed or used up. It is just staying as magnesium. It's just there to just stabilize the reaction and make it more possible. So as I mentioned before, this is a stepwise phosphorylation process. So we're at step 1 now. So we've got our monophosphates. Okay, so now we need to go from a deoxyribonucleoside monophosphate to deoxyribonucleoside diphosphate. Okay, so we want to add an additional phosphate group now to the phosphate group that's linked to the deoxyribose sugar. Okay. Now, the enzymes that do this respectively, so we've got our deoxyadenosine monophosphate here, the kinase enzyme that catalyzes that phosphate transfer between ATP and DAMP is called adenylate kinase. For deoxyguanosine monophosphate, it's guanylate kinase. For deoxycytidine monophosphate, it's cytidine monophosphate kinase. and for thymidine monophosphate, it's thymidylate kinase. Okay, and as I said, the end product will be a deoxyribonucleoside diphosphate. So, for example, let's take deoxyguanosine monophosphate. With the help of guanylate kinase and ATP, it will become deoxyriboguanosine diphosphate. So um, we're now indicating that there's another phosphate on to, as well as that additional that original phosphate that was added on in the step 1 phosphorylation process. So again, I just kind of thought I'd highlight this kind of step 2 process just so we can really understand what's going on per nucleoside monophosphate to nucleoside diphosphate. So we're using the example of adenine here. So this is deoxyadenosine monophosphate here. You can see this is the adenine nitrogenous base. We've got the imidazole ring, and you can see that nitrogen on the imidazole ring is linking that deoxyribose sugar. That's the glycosidic bond, and you can see this deoxyribose sugar is linked to this phosphate group via a phosphoester bond. Okay, and essentially our goal is to get deoxyadenosine diphosphate from deoxyadenosine monophosphate. So we need to add another phosphate. Okay, we need to phosphorylate it again. Again, we need the help of ATP to act as a phosphate donor, and we still need magnesium as a cofactor. Again, because this hydroxyl group that we want to attach another phosphate to is very negatively charged. Hydroxyl group is another functional group, and again, I'm going to go over functional groups in later videos and also in part 2 of this video. But it's negatively charged, and so are the beta and gamma phosphates. The oxygens on those phosphates are negatively charged in ATP, so there's a natural repulsion there. So we need some kind of like charge shield. We need some kind of stabilizer, and that's where magnesium comes in. Okay, and with the help of adenylate kinase, that phosphate group is transferred, and the deoxyadenosine monophosphate becomes phosphorylated to deoxyadenosine diphosphate. And once again, my head's covering ADP, but ADP, adenosine diphosphate, is a byproduct to be expected because adenosine triphosphate has just lost a phosphate group, so it becomes a diphosphate. Okay, and it's instead of a phosphoester bond being formed, it's a phosphoanhydride bond being formed that is between this phosphate group and that phosphate group. Okay, so between, we'll call it the alpha phosphate and the beta phosphate. The beta phosphate's been attached to the alpha phosphate, and what links those two together is a phosphoanhydride bond. So again, just to recap, I'll demonstrate the same for guanosine monophosphate. So we've got our deoxyguanosine monophosphate right here. You can see that this is guanine. You can see it's got that carbonyl group on the 6th carbon in the pyrimidine ring. You can see that the imidazole ring forms the nitrogen in the imidazole ring forms the glycosidic bond with the deoxyribose sugar, and then you can see this phosphoester bond here to form this is our alpha phosphate. So still negatively charged. It's got that negatively charged hydroxyl group. Okay, so ATP still adenosine triphosphate ATP is still acting as our phosphate donor. Okay, and magnesium is still stabilizing that charge repulsion. Its positive charge is stabilizing that between the beta and gamma phosphates in the ATP and the hydroxyl group on this phosphate. Okay, and with the help of guanylate kinase to catalyze this reaction, additional phosphate group is added or transferred from ATP to give us deoxyguanosine diphosphate. Okay, and as a byproduct we get adenosine diphosphate. Okay, because ATP's lost a phosphate group. And again, it's important to know that the bond formed between the alpha phosphate and the beta phosphate on the deoxyguanosine diphosphate, it isn't a phosphoester bond like it is with the alpha phosphate and the ribose sugar. It's a phosphoanhydride bond that is formed between these phosphate groups. So we'll just go through deoxycytidine monophosphate now, just briefly. So you can see I've started off here in our equation. We've got deoxycytidine monophosphate. There's just one phosphate group. Again, you can tell it's cytosine because we've only got one carbonyl group, and then there's the amine group. You can see it's just a pyrimidine ring. There is no imidazole ring like there would be if it was adenine or guanine as a nitrogenous base. Okay, and that nitrogen in the pyrimidine ring is linking with this deoxyribose sugar, and then we've got our phosphoester bond. Okay, and with the help, once again, ATP is really important here for phosphate transfer. Okay, and again with the help of magnesium to stabilize that kind of repulsion between the beta and gamma phosphates in ATP and this negatively charged hydroxyl group. this will allow for this will make it easier to catalyze for citidylate kinase to catalyze this reaction and essentially transfer that phosphate group, and then we get deoxycytidine diphosphate. Okay, so there's now two phosphate groups, and then remember we've formed a phosphoanhydride bond. Okay, we haven't formed a phosphoester bond, we formed a phosphoanhydride bond. Okay, which is between the alpha and beta phosphates. Okay, the phosphoester bond is formed between the alpha phosphate and the deoxyribose sugar itself. So last but not least, we've got our thymidine monophosphate, okay, and this is the stepwise phosphorylation from thymidine monophosphate to thymidine diphosphate. Okay, so we start off with our thymidine monophosphate. As you can see, we've got our thymine nitrogenous base. You can see it's got the two carbonyl groups that cytosine doesn't have, and the methyl group is here, although it's not indicated in this diagram. Okay, it's linked on the nitrogen in the pyrimidine ring to the deoxyribose sugar through a glycosidic bond. We've got our phosphoester bond here, and we've got our negatively charged phosphate group. Okay, now ATP once again acts as a phosphate donor, and magnesium again, it stabilizes that repulsion, and there isn't a hydroxyl group here, okay, but it doesn't matter because oxygen is still very negatively charged, so it still will naturally there'll be some level of repulsion between the beta and gamma phosphates on the ATP and the phosphate that's on this thymidine monophosphate. Okay, so that magnesium is still acting as some kind of stabilizer, and with the help of thymidylate kinase, which catalyzes this reaction, it essentially transfers the phosphate group, right? It makes the reaction possible. You get thymidine diphosphate. So we've gone from thymidine monophosphate to thymidine diphosphate, and of course my head's once again covering it up, but you get ADP, adenosine diphosphate. Okay, and once again it's important to know we've formed a phosphoanhydride bond here. Okay, not a phosphoester bond, a phosphoanhydride bond. Okay, and we've now got our alpha and beta phosphates on our thymidine diphosphate. So now we're on to step 3. So we've now got our nucleoside diphosphates. Okay, so they need to be phosphorylated one more time. As I said, it's a 3-step process. Okay, but this step is a lot easier to remember in the sense that the enzyme that catalyzes this transfer, this final phosphorylation from ATP to the nucleoside diphosphates, is a universal kinase enzyme, which means it's the same enzyme that does it for all 4 of the nitrogenous bases, that's called nucleoside diphosphate. So just to recap, for example, we've got deoxyadenosine monophosphate. Okay, in order to the enzyme that catalyzes that phosphate transfer from ATP is adenylate kinase. Conversely, for guanosine monophosphate, to get guanosine diphosphate, you use guanylate kinase to catalyze that transfer of phosphate groups. Okay, but so there's different enzymes, but for this process it's the same enzyme. It's nucleoside diphosphate kinase. Okay, now we've now got 3 phosphate groups on our nucleoside, so it's now a deoxyribonucleoside triphosphate. Okay, so we've got an alpha phosphate, and the alpha phosphate is linked directly to the deoxyribose sugar through the phosphoester bond, and then the alpha phosphate is linked to the beta phosphate via a phosphoanhydride bond, and the beta phosphate is linked to the gamma phosphate via a phosphoanhydride bond. Okay, now why do we need to phosphorylate 3 times in order for DNA to polymerize? Because a DNA mononucleotide is just a nitrogenous base, the sugar, and 1 phosphate group. So why 3? Well, polymerization, it requires, to put it simplistically, and we're going to go over this a lot more when we look at the significance of phosphate and energy and all that stuff, it provides the energy release allows for the polymerization to occur. So let's say when 2 or 3 or 4 triphosphates are coming together to form that kind of polynucleotide, so when all those monotriphosphates are coming together, if you like, to form that polynucleotide, what's happening is that those 2 phosphate groups are being released. Okay, they're being released, so you get pyrophosphate, okay, PPI in equations is being released, and that release of the beta and gamma phosphates allows for the it allows the energy, it provides the energy for that work to happen, that polymerization for DNA polymerase enzyme to to polymerase that, okay, to put that form that polymer from those subunits. Okay, so the energy stored in that bond allows for this to happen, and you can see that with ATP as well. Notice ATP always has free phosphates, okay, because the energy stored in those bonds, especially when either the gamma or beta's gone, it's cleaved off. It allows for free energy for work to happen, essentially. But we'll go over that more later. So I'll just use a deoxyadenosine diphosphate as an example. I won't go too much into the step 3 because we kind of get the idea, and it's the same enzyme that will catalyze this transfer of phosphate groups, okay? But okay, so we start off with our deoxyadenosine diphosphate. So once again, just to break it down, we've got our nitrogenous base. You can see that's a purine. You can see it's adenine because on its 6th carbon it's got that amine group, whereas guanine would have a carbonyl group, C double bond into an O. Right, you can see the imidazole ring is forming that linkage, that glycosidic linkage with the deoxyribose sugar, and you can see this phosphate group is linked to the deoxyribose sugar through a phosphoester bond, okay? And then this is our alpha phosphate, and it's linked to our beta phosphate via a phosphoanhydride bond, okay? There's a lot of high energy stored in this covalent bond, okay? So when it cleaves, it's gonna allow for work to happen, essentially. Right, so then we've got ATP again is our phosphate donor, and we've got magnesium as a cofactor to stabilize that repulsion between the beta or between the gamma phosphates on the ATP and the this hydroxyl group on the diphosphate, on the deoxyadenosine diphosphate. Okay, so with the help of nucleoside diphosphate kinase, which, as I mentioned before, ideally should appear above this arrow, but I have no idea how to get that to happen. PowerPoint was not working for me today. We'll catalyze that transfer of, or catalyze that phosphorylation, okay, and essentially we'll get deoxyadenosine triphosphate. So we've now got 3 phosphate groups. Okay, we've got our alpha phosphate, which forms the phosphoester bond with the deoxyribose sugar. We've got our beta phosphate, which forms a anhydride bond with our alpha phosphate, and then we've got a gamma phosphate, which forms a phosphoanhydride bond with our beta phosphate. And then as a byproduct, we'll have ADP, adenosine diphosphate, but once again, my head is covering up that plus ADP, but it's really important to include it in the reaction. Okay, and as I said before, the enzyme that catalyzes this for all 4 different reactions is a universal enzyme called nucleoside diphosphate kinase. Now, I use the term universal enzyme very loosely. I don't particularly think it's a formal term. It's just a term that a lot of biochemists use, okay, in this context, because it's not a specific enzyme. This enzyme's quite flexible. It's able to catalyze the transfer of that phosphate from ATP to the nucleoside diphosphate for all four reactions. Okay, it doesn't matter if it's deoxyadenosine or deoxyguanosine or thymidine. Okay, it will work on all 4. Okay. So I just thought I'd kind of dedicate a slide to magnesium's role in these processes, and we'll be talking about cofactors a lot more as we progress on throughout these biochemistry videos. But for now, we're just going to emphasize the importance magnesium plays in nucleotide metabolism, and I think it's really important to indicate magnesium's involvement when you're drawing out mechanisms or equations. It's really important to indicate magnesium's involvement. Okay, so the diagram I'm showing on this slide is adenosine triphosphate, or ATP. As everyone knows, it's such an important molecule in metabolism. It's the primary candidate responsible for almost all the phosphate donations, for phosphorylation, and that phosphoanhydride bond that covalently links those 2 phosphate groups, especially the gamma and the beta, right on the end, there's a lot of energy that's stored in those bonds, and when they're cleaved or released, it releases a lot of energy for allow a lot of reactions to become feasible. Now, to put it into context, they're naturally negatively charged. You can see that's indicated on the oxygens. Oxygen's naturally very negatively charged, and we'll look at this a lot more when we look at functional groups. We look at their role in hydrogen bonding and stuff, but for now, just just understand the fact that these oxygens are very negatively charged. And let's say we've got our adenosine nucleoside, okay? So our adenosine nucleoside is just a nitrogenous base, and it's just a deoxyribose sugar. There is no phosphate group yet, right? But the 5' hydroxyl group is also negatively charged, and that's the functional group that will eventually come on to form that phosphoester bond between the deoxyribose sugar and the phosphate itself. Okay, so we want to get a nucleoside monophosphate. We want adenosine, deoxyadenosine monophosphate from just adenosine, deoxyadenosine. Okay, but for this reaction, of course, is catalyzed. It's catalyzed by the adenosine kinase, really important in catalyzing and making that reaction feasible. But magnesium also has a really important role, because the hydroxyl group on that sugar is going to naturally be repelled by these oxygens on this phosphate here. So there's some level of electrostatic repulsion, if you like. So that's where magnesium comes in. You can see it's indicated as a 2+, right? It's positively charged. Okay, so it's acting as a charge shield. It's preventing, or it's reducing that repulsion. It's making the reaction become more feasible. Okay, so it's important to know though that the magnesium's not directly acting on the active site of the enzyme. Okay, its role is to stabilize the negative charges, hence it's not consumed or used up. It's not changed, which is why we're not including it on the as a product. Okay, or it's not changed or consumed or used up, but it's reducing that electrostatic repulsion between ATPs, beta and gamma phosphates, and either the hydroxyl group that's on the deoxyribose sugars on the nucleosides, or even when you look at, say, adenosine or deoxyadenosine monophosphate, that contains one phosphate, but that hydroxyl group on that phosphate group is also going to be negatively charged. The same with a different example would be the outlier here would be thymidine, for example. Thymidine monophosphate or thymidine diphosphate, instead of a hydroxyl group, is going to have that O-. They're still negatively charged. So ATP is always acting as our phosphate donor, but it's without magnesium, it's going to be repelled. Okay, the gamma phosphate is going to be repelled. So magnesium will stabilize that, right? And it allows for a correct positioning to allow the phosphate transfer, right? It stabilizes the enzyme substrate complex. That's a really important key word, okay? And it may come up in exams. It's the role of cofactors like magnesium is to stabilize the enzyme substrate complex, right? And this will allow the enzyme to bind to its substrate, right? It stabilizes that reaction, and it reduces the repulsion between the phosphates, the phosphate donor, and the phosphate acceptor. Okay, it's reducing that repulsion, it's stabilizing that reaction. And you can see that here. I mean, you can see magnesium is a 2+ charge, and we've got an overall 2- charge here from our oxygen. It's acting as a charge shield. So just to recap, that magnesium is acting as a cofactor. It's stabilizing that enzyme substrate complex by reducing the electrostatic repulsion between the phosphate donor and the phosphate acceptor. So now we're going to move on to the actual polymerization of DNA itself. And the enzyme that catalyzes this reaction is referred to as DNA polymerase. And a lot of people will recognize DNA polymerase when looking at DNA replication before cell division, but we're not really going to worry about that too much. We're just looking at it purely from a chemical process, what's actually happening. So I've got an equation up here, and not to be intimidated, we'll go step by step and we'll break it down. But I've got DNA brackets N OH. Now, what does that mean? So the DNA brackets N refers to the polymer of DNA. So we don't know, as we know, DNA is a polymer, it's a polynucleotide, and it's made up of many mononucleotides, right? That make that long strand. So we're not specifying how many there are, but there are enough for it to be considered a polynucleotide. So a lot. Okay, so it's just signified as N. But then we've got this OH. Now this stands for the hydroxyl functional group, okay? And this hydroxyl functional group is on the 3'. It's the 3' hydroxyl group on the growing DNA strand. This hydroxyl group is what we're interested in. This hydroxyl group will go on to form that phosphoester bond between the phosphate on an incoming nucleotide, on an incoming mononucleotide, right? And essentially that phosphoester bond is vital right, in polymerization. Okay, but once again, that as we know, hydroxyl groups are negatively charged, right? So again, we need magnesium to kind of stabilize that. But to not get ahead of myself, I've just said DNA N OH. So that refers to the polymer of DNA and the 3' hydroxyl group that's coming out, that's part of the deoxyribose sugar. Okay, and then we've got our deoxynucleotide triphosphate. Okay, so that can either be deoxyadenosine triphosphate, deoxyguanosine triphosphate, thymidine triphosphate, or deoxycytidine triphosphate. Okay, so that is what we want to add on to this. Okay, and this arrow here, again, I really apologies for my PowerPoint or lack of PowerPoint skills. Okay, I don't know how to put a DNA polymerase to indicate on that arrow, but it's really important, right? This arrow here, DNA polymerase, goes there because that's the enzyme that is actually making this reaction possible. Okay, and this gives us DNA N+1. So we've just added, N+1 refers to the mononucleotide that has been added onto that polymer. Okay, it's polymerization. Imagine we're assembling all these nucleotides to form a polynucleotide. Okay, so we've got our N+1, but we've still got that hydroxyl group. It's really important to indicate that hydroxyl group because that will allow for another nucleotide to join on, and then so again, and then so again, plus pyrophosphate. Now, that's PPI. Now, the pyrophosphate, you might be wondering, where has that come from? Well, that's come from the deoxynucleotide triphosphates. Okay, so if we go back a few steps, right, we know that DNA is a polynucleotide. It's made up of many mononucleotides, and a mononucleotide consists of a nitrogenous base bonded to a deoxyribose sugar, and that deoxyribose sugar is linked to a phosphate. Okay, so there is only one phosphate, but in this stepwise phosphorylation process, we phosphorylated our nucleoside, our nitrogenous base, and our deoxyribose sugar. We phosphorylated that 3 times. Okay, so we don't actually need those 2 phosphates, not in the core structure of DNA itself, right? But those 2 phosphates are really important, right? Because one DNA polymerase requires them to work, right? It requires them to polymerize DNA. And a lot of that's to do with the I won't get into it too much, but as I mentioned before, the covalent bonds, those phosphoanhydride bonds, they store a lot of chemical potential energy, I guess, if you like. And that cleavage of pyrophosphate being released allows for free energy essentially for the work to happen, right? Okay, so there's a lot of important things to remember. Okay, so we've got pyrophosphate as a byproduct. Okay, pyrophosphate is clean, but we're only really interested in this alpha phosphate here. This beta and gamma phosphates used for enzyme recognition. Okay, but also to allow for energy essentially. But now we're going to go back to the role of magnesium. Now I haven't actually included magnesium in this equation, but I think it's important to include it. But I'll still talk about it. As I said before, the magnesium is stabilizing that. It's still acting as a charged shield, okay? Because we've got a negatively charged hydroxyl group, okay? All right, that's electronegative, if you like, overall, right? So there's going to be some form of electrostatic repulsion between this gamma and beta phosphates that are on this triphosphate nucleotide. Okay, so we need magnesium to act as a stabilizer, right, between the 3' hydroxy group and the negatively charged triphosphates, okay? And this essentially allows, this magnesium itself allows for a nucleophilic attack from the 3' hydroxyl group that's on the deoxyribose sugar and the alpha phosphate efficiently. So it allows for that phosphodiester bond to be formed. It allows for that phosphate sugar linkage, or that phosphoester bond, to be formed. So just to break the equation down again, we've got DNA ends, we've got a DNA polymer that has a 3' hydroxy, okay? That's what hydroxyl group. That's what we want our nucleotide to bind to for a phosphoester bond. We want that alpha phosphate to bind to this hydroxyl group, okay, to form a phosphoester bond to allow for the strand to continue to grow. It allows the strand to continue to grow. Okay, so we've now got DNA, we've got our DNA strand, we've added one nucleotide onto it, and we've got that hydroxyl group on the deoxyribose sugar of the nucleotide we've just added, the 3' hydroxyl group, to allow DNA polymerase to add another nucleotide base onto that, and as a byproduct we've got pyrophosphate. Okay, and the energy that's gained, that free energy that's gained from cleaving that gamma and beta phosphate, allows the work to happen. Okay, and magnesium's acting as a charge stabilizer. Okay, in this process too, it's preventing that repulsion between the hydroxyl group and the beta and gamma phosphates on the triphosphate nucleotides. So, okay, I'm just gonna walk through this kind of diagram I've got down here as well, just to kind of build a picture again, just to reinforce this concept. So you can see here we've got a growing nucleotide polymer, and we've got our 3' hydroxyl group, okay, and that triphosphate nucleotide, we've got our alpha, beta, and we've got our gamma respectively, but the alpha is what's going to form a phosphodiester bond with this hydroxyl group. Okay, magnesium's there acting as a charge stabilizer and stabilizing that enzyme substrate complex with DNA polymerase, and it's allowing for that binding. Okay, and then what's happening is pyrophosphate or diphosphate is more commonly referred to as diphosphate, but it can also be referred to as pyrophosphate, right, is being given off. Okay, and we're getting that as a byproduct. Now there's now 4 nucleotide monomers. We started off with 3 and we added 1, and then this here there's going to be another hydroxyl group coming out of that purple pentagon, that pentose sugar, that 5-carbon sugar, to allow this process to happen again. And DNA polymerase will essentially run, and it will run across that. So for example, during DNA replication, it will run across that template strand, and it will polymerize accordingly. It will form an adjacent strand, right? And now, just to talk about this as a polymer, right, some important points to remember. Okay, the phosphoanhydride bond is broken off, and free energy is allowed for work to happen. Magnesium stabilizing the electrostatic repulsion between the 3' or the 3 hydroxyl groups, allowing for a nucleophilic attack from the 3' hydroxyl and the alpha phosphate efficiently, and the phosphoester bond is formed. When you're looking at DNA as a polymer, though, it's important to remember that it's a phosphodiester bond, because if you look, we've got in purple you've got your sugar, okay, and then in orange you've got your phosphate group, and you can see here these phosphates actually they're responsible for bringing 2 monomers together, if you like. That phosphate's able to form a phosphoester bond with a neighboring deoxyribose sugar on a neighboring nucleotide monomer. Okay, so that's therefore referred to as a phosphodiester bond. So when it's a mononucleotide, when you look at a nucleotide monomer, you'd refer to that sugar phosphate linkage as a phosphoester bond. But when you're looking at a strand of DNA, you'll notice it's considered a phosphodiester bond, because that same phosphate is allowing for linkage with the hydroxyl group on another sugar. So it's essentially it forms another phosphoester bond, so it's a phosphodiester bond. So that's essentially the process of DNA polymerization, to put it simplistically. The next part 2 is going to cover hydrogen bonding, complementary base pairing, and I guess the directionality of DNA. It's 3' to 5'. We're going to be looking at sense and anti-sense strands. We're going to build on from there. But that's the end of this lecture. Well done if you made it this far. And yeah, I wish you the best of luck in your studies.